Hello and welcome, dear students. How are you all? So I guess all of you have been waiting since long. Dear students, please report your attendance. The class is starting right away now. Students, please report your attendance. Okay. So dear students, please report your attendance. Class is starting now. Can you all hear me? Okay. All right. So thing is, can you all hear me now? Hello everyone. Yeah. I hope you can hear me now. Okay, great. So Tamenglong students, how are you? And Shillong centers, please report attendance. Itanagar, Shillong, Tamenglong, and yeah, Chura Chandpur. Please report your attendance, and we are going to start right now. Okay. So sorry for that inconvenience that you had, and I hope my sound is visible to you. Sorry, sound is audible to you now. All right. So in the last class we did quite a lot of things. We were discussing about how there are a few questions related to the arrangement of xylem, the arrangement of protoxylem and metaxylem specifically. So regarding that, I have to talk to you. I also gave a bit of homework to you in the last class. Have you done it? Have you done your homework? Yes or no? All right. There was only one question that might have been difficult for you regarding endarch and exarch. Endarch and exarch condition of xylem. So please settle down quickly. Please settle down quickly. So that might have troubled you for a bit. Endarch is a very important question for your PMTs. Endarch is a condition in which protoxylem is in the center and metaxylem is in the periphery. This is endarch condition. So if I draw xylem like this, if I draw xylem vessels like this, larger vessels which will be metaxylem vessels towards the periphery. Okay, my voice is echoing. All right. I'll try to be a bit slow. All right. So I hope this works for you. Protoxylem towards the center, metaxylem towards the periphery. Okay. So I cannot help right now with the echo. I will try to speak more slowly. Protoxylem Px, metaxylem Mx. Okay. So this is the endarch condition. You can write the arrangement. I will speak very slowly so that it does not echo. The arrangement in which protoxylem, this is your homework that I am discussing, in which protoxylem is present at center and metaxylem towards periphery is called endarch condition. Okay. So this is endarch, and the opposite of this condition, it is exarch. Okay, my voice is echoing again. I'll try more slowly. Okay. Exarch. Exarch is the arrangement. Exarch is the arrangement in which protoxylem is towards periphery, protoxylem is towards periphery and metaxylem towards center. Metaxylem towards center. Students, I'll try to write more because I believe my voice is echoing still. Okay, I'll try to speak less and write more. Okay, so this is exarch condition. This is the center. We are moving towards periphery. Yeah. Okay. Please keep on checking some messages. They are from the technical team, for the technical team. Okay. I'll keep writing in between. Protoxylem is towards periphery. Towards the center, it is metaxylem. Here it is protoxylem. Okay. So, do you remember where we have seen this condition? Okay. All right, you remember this condition, yes or no? All right. So, here protoxylem was towards center, and metaxylem is towards periphery. So, if I will take a look at the, you cannot hear me, all right. There is a problem that you cannot hear me. Can you hear me now, Senapati, dear students? Yeah. Please check if you can hear me, yes or no. Can everyone hear me? Okay, I am writing this message, I am not sure because, all right. You can hear me a bit. So, is my voice not loud enough, I suppose? Okay. All right. So, Tamenglong can hear me, Shillong students can hear me, but I suppose there is a problem. Okay, it's the echo. Shillong is having the echo. All right. Please keep on checking the messages. Our technical team is there. So, in the meantime, I am going to draw some diagrams until this problem is fixed. So, I'll try to speak bit less. Right. So, today what we have to do is, we have to do leaf structure, okay. Leaf dicot and monocots. Okay. So, we are going to begin the drawing the transverse section of the leaf of a dicot leaf first and the monocot leaf later. Right. So, Ts of dicot leaf. I am speaking a bit less. I am going to focus on the diagram. So, in the meanwhile, this problem I hope will be resolved. Piece of dicot leaf. All right. So, please draw with me. Okay. Wait a bit. So, draw it like this. This is the upper surface of leaf. This layer I am drawing it is the cuticle. 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 Now, just below this are going to be some very huge number of cells. Okay. Shillong, you have a message for the technical team, for your coordinator. This is not for the students, not for the students. All right. Now, you have to draw column shaped cells like this. Long elongated cells like this. You have to draw this. Okay. This is what I am drawing here. Long elongated cells. After this, we are going to draw a special structure called the bundle sheath. Okay, so this is called the bundle sheath, and inside this they are going to be present some xylem vessels. So, I am going to draw them with blue color, blue like water. They are going to be conducting a lot of water and minerals. So, this is I am going to draw it with blue color. Xylem vessels are present in leaf also. The remaining part I am going to fill with phloem cells. These are phloem cells. As you know, students, I am always using red color to draw phloem, right? Like this. Please fill all the space between bundle sheath and xylem with red colored or any color of your choice phloem cells. Okay, these are phloem cells. Now, the remaining part for that first these long cells, these cells, long cells like this. These are the palisade mesophyll cells. Now moving on towards the lower epidermis of the leaf. So I am going to draw the lower epidermis. Lower epidermis. Like this. Okay. Like this. Okay. All right. And in the rest of that space, let me fill these cells which are the spongy mesophyll cells. So I am going to draw slightly less green colored large cells with huge amount of intercellular spaces. Huge amount of intercellular spaces. Like this. Like this. Okay. Like this. So these are going to be called the spongy mesophyll cells. Okay, these are the spongy mesophyll cells. Okay, so I am going to draw now the other cells which are going to fill this space slightly more dense than spongy mesophyll cells, like this. Okay. Now the structure of leaf is complete. So let us label now. Cuticle is done. Adaxial surface. Look, upper surface of leaf is called adaxial surface. So this is the adaxial epidermis. Adaxial epidermis. Now this part, it is called the air cavity. Okay, this is the air cavity. This is the spongy mesophyll. Okay, this is the stoma. Okay, this is the abaxial epidermis. Okay, and This of course is the xylem. This is the xylem, and this is the phloem. Okay, this is the phloem, and what remains now? Yeah, now it is the bundle sheath. Bundle sheath. Bundle sheath. Okay, and spongy mesophyll is labeled. Okay, one cell is still remaining. This is the column shaped cell that I draw earlier. This cell is the palisade mesophyll cell. Palisade mesophyll cell. Okay. All right. So this is yeah, cuticle, upper cuticle, lower cuticle. This is also the cuticle. Cuticle. And these cells are the guard cells. I suppose you know, everybody, right? So finish the diagram. So is my voice still echoing? Okay. Shillong students, is my voice still echoing? Okay, great. Yeah, I'm very happy that it's been resolved. All right. So what we have done here, there are two surfaces of the leaf. This is a dicot leaf, and everybody sees that when you see a dicot leaf, the upper surface is more green in color. You know, the upper surface is more green in color, and the lower surface is slightly less green in color. So that's because of in the lower surface the cells which have chloroplasts, these are the spongy mesophyll cells, and they have large intercellular spaces between them. And one more thing, Senapati, you cannot hear anything. All right. So okay, yeah, our technical team is working on it. There have been some issues, so our technical team is working on it. Senapati, if you hear anything, please let me know. Our technical team will contact you shortly. All right. Okay. All right. So please wait. All right, so we are working on it. Okay, so for the other people who can hear me, this lecture will be uploaded also, so you can pick it up from there. In the meantime, those who can hear me, I'm just explaining the things now. When you have to draw a leaf, you will draw the stomata only on the lower surface, only on the lower surface of the leaf, not on the upper surface. The upper surface, the stomata are nearly absent or very few in number. You can see here in this diagram, I have not drawn any stomata on the upper surface. On the adaxial surface, no stomata will be present, or very few will be present. It's better to not to draw any stomata, okay, or not to say that there are stomata at upper surface. If there are, there will be very, very few in number. Most stomata or all stomata are located on lower leaf surface. Can anybody tell me the reason why? Can anybody tell me the reason why? because there has been a lot of issues, I won't take much of your time. After 3 o'clock, I'll leave you, okay, because I think there are a lot of issues today. So don't worry. I will take very few things. I'll just try to finish this chapter somehow, and in the next chapter, I will deal in the next class. So up till then, please have some patience. We are really working on it. I am not sure what went wrong, because that's the technical team's issue. They are working on it. There might be a satellite uplink issue or downlink issue. So okay, we are working on it. Please be patient. All right. So why in this leaf surface, how you have to draw it? Upper surface you will not draw any stomata. On the lower surface you will draw stomata. On the upper leaf surface, long elongated cells are present, which are not present on the lower leaf surface. In this diagram, you will see that there are not any long elongated green colored cells on the lower surface. They are only on the upper surface. They are very tightly packed, 1, 2, 3, 4, 5, 6, very tightly packed, and they are column shaped, okay, like pillar shaped cells. So these are called palisade cells. Palisade means pillar like. Below that is the stomata. Stomata has the function, okay, yeah, very good, Shillong. Stomata are not present on the upper surface, because if they will be present, there will be large amount of water lost from the upper leaf surface. Why? Because wind is also blowing, and the sunlight is also falling on the upper leaf surface more compared to the lower leaf surface. So there will be high transpiration. Okay, that is the thing. So that is why dicots have this adaptation. They will place their stomata on lower leaf surface. All right. Okay. So in the lower leaf surface, there are spongy mesophyll cells which allow the amount of air which has been getting inside from the stomata to circulate in the whole leaf area. In the whole leaf volume, this air will be circulating, because leaf cells also respire. So they need oxygen. Without oxygen, they will die. Also, they make oxygen out of carbon dioxide. They make oxygen out of carbon dioxide and water. Both things are required. Actually, oxygen comes from water, but this oxygen must be released quickly. If it is not released, more food will not be formed. So they need to quickly circulate this air very fast. All right. So stomata has to be open, and air has to circulate among the cells. Then only these processes will happen. Okay. So this is the thing. This is the dicot leaf structure. Now after this, we will draw the transverse section of monocot leaf. One more thing, here a particular type of cell is present, which is called the bundle sheath cell. These cells, these green colored cells, which have now become black suddenly. Okay. So these are the bundle sheath cells. I have shaded slightly more area than I was trying to, but I hope you get the idea. Okay. So these cells, these, okay, if I go, yeah, like this, these cells now, these are the bundle sheath cells. Okay, it's clear now. These are the bundle sheath cells. Black and green cells, they are the bundle sheath cells. They surround this whole vascular structure in the leaf. These bundle sheath cells will be of great use when I teach to you the C4 cycle of photosynthesis. So I will remind you that there is a thing called bundle sheath cell when I teach you this part. Okay. So later I will come up to this. Okay. So this is for the dicot leaf. Let us move on to the TS of monocot leaf. Okay. Senapati, can you hear me now? Senapati, you can hear me now. This lecture will also be uploaded. Okay, so don't worry about missing out. Can you hear me now? So you can reply on the chat. Okay, so I hope this is resolved now. All right. So now I will move on to the transverse section of dicot versus monocot leaf, and I will explain to you the differences. All right. So everybody can hear me now. Fine. So please put the heading transverse section of monocot leaf. Look, a monocot leaf is called isobilateral leaf. Isobilateral leaf. Whereas a dicot leaf, it is called dorsiventral leaf. Dorsiventral leaf. A dicot leaf is dorsiventral, meaning there is a dorsal surface and a ventral surface. A dorsal surface and a ventral surface. But in monocot leaf, suppose you are looking at a grass leaf or a bamboo leaf. So both the surfaces are exactly the same. Whereas a dicot leaf, if I give you a dicot leaf, you will be able to know that this is the upper leaf surface and this is the lower leaf surface. There is a huge amount of difference between these two surfaces. You know very well that this is the upper, this is the lower. But in monocot leaf, it might be a bit difficult because both the surfaces look exactly the same. A bamboo leaf, a sugarcane leaf, a wheat leaf, rice leaf. Are you getting me now? So this is the thing. So a dicot leaf is having distinction between upper and lower leaf surfaces. Monocot leaf, it has stomata on both surfaces and also both the surfaces are looking like each other. So that is why we call it isobilateral leaf. No distinction between upper and lower surfaces. Alright. So let's draw it now. It's quite easy compared to the dicot leaf. No such bit of an issue. Alright, so let's draw. We'll draw stomata here on the both surfaces. Okay, we'll draw stomata on both surfaces here. So this is the first stomata on the upper leaf surface. Okay, wait a bit. Okay, so I am drawing a bulge like surface here. The reason is, okay, wait a second, this is not perfect. Like this. Yeah, now it's fine. Yeah. So this is the upper leaf surface. The lower leaf surface is less complicated. We do not have to draw any bulges here. We will draw almost like a straight line. Like this. Like this we will draw. Okay. All right. So coming to the inside, we will draw like spongy mesophyll cells. These cells are also here, but the thing is, these cells do not have any intercellular spaces. This is the difference. So we will draw cells here, but we won't draw any large intercellular spaces here. Just normally like that we draw it. Also, here there is a patch of sclerenchyma, here, and between these xylem vessels will be present like this. So, you can draw it like this. These are the xylem vessels. Okay. The remaining space, okay, wait a second, I used to draw it with blue color. I must draw it with blue color because if it's my convention, I must follow it. Alright. Xylem I am drawing with blue color, phloem I will draw with red color because outside is all phloem, so it will be bad if I don't use that convention. Now I can use the rest of this space to fill it with phloem. The rest of the space we will fill it with phloem. Okay, like this. The rest of the space you will fill it with cells like this. Like this. Right. So, pink colored cells, these are actually sclerenchyma patches. These are basically sclerenchyma patches present just above and below the vascular bundles. They are just there to strengthen the vascular bundle, nothing more, nothing less. Like this. Now, the remaining space we will fill it with spongy mesophyll cells. So, basically we will not call it spongy mesophyll, but just like that, large cells, tightly packed cells. Large cells, tightly packed cells. Like this. The rest of the space also we will fill it like this. Like this. Basically, right. So, this is the isobilateral leaf. This is the isobilateral leaf. Stoma. This is the abaxial epidermis, abaxial or lower epidermis. This is the xylem, this is the phloem, this is the mesophyll. Okay. So I guess this is pretty much enough. Yeah, this is the adaxial epidermis. Adaxial epidermis. This will be the cuticle. This will be the cuticle, like this. Okay. So this is the monocot leaf structure. So this is about the monocot leaf structure. Now dicot leaf and monocot leaf, they are very easy, but in monocot leaf there is a bit of a problem. Here there are a large amount of stomata on the upper surface also. So how do we resolve this problem? This is going to be a problem for the plant. The diagram please. Yeah. So there is going to be a bit of a problem for the plant. The problem is, you please keep on drawing the diagram. Yeah, please keep on drawing the diagram. So the thing is, okay. Everybody, as soon as you finish the diagram, please let me know. Okay. So if there are stomata on both the leaf surfaces, a large amount of water will be lost. Okay. So for those plants which are crop plants like wheat, rice, okay, maize, so you must have seen that these plants require large amount of water. Anybody who has seen the farmers growing wheat, growing rice and sugarcane and maize, do you remember that how much water do these plants require? If you have seen a crop land somewhere growing these monocots, do you understand how much amount of huge quantities of water that is required? Say yes or no, if you know about it. If you don't know about it, you can ask the farmers who grow these crops. Okay, you have not seen? Okay, yeah, some people have seen. Yeah, so these require so much water that actually the farm is actually got to be flooded with water sometimes. So because they undergo huge amount of transpiration, if we do not give water, these plants will not be able to give the fruit, and their fruit is caryopsis, the wheat seed, and that is what we consume. Rice is also caryopsis, maize is also caryopsis. So these plants require huge quantities of water. But there are some grasses in which do not have such large quantity of water access. Okay. So the grasses have spatial adaptations. Their adaptation is like this. Suppose you have a piece of paper, okay, and you have a piece of paper and you want to fold it, but if it has been already folded once, so there are some lines on the paper, okay, so it can easily undergo folding in that direction, on the direction of the lines, right? Similarly, in grasses there are some cells, and these cells have huge volume cells. They can easily lose water. When they lose water, they form lines, they form creases on the leaf surface, for which the leaf surface will quickly start to fold. Okay. Everybody has some lines on their hands, right? Everybody has some lines on their hand surface, right? So these lines represent the pattern. When you were first born, you had folded your hands in some particular pattern, and these lines that are on your hand, these represent those patterns. Okay. So when you fold your hand like this, you can see like along these lines your skin will start to fold, right? Because there is a huge amount of skin on your palm surface, hand palm, just like this. So, their leaves also have some foldings, creases around which these leaves will quickly fold. So, if this is a very important question for NEET exams. What are these cells which help in leaf folding? When the leaf will fold, it will reduce the leaf surface and it will also reduce the transpiration. I hope you got me. So, please write now. Monocot leaf, there is nothing special about it. You have already seen the diagram, but you can write a few points. In monocot leaf, stomata are present on both leaf surfaces. Stomata are present on both leaf surfaces. The stomata is also not differentiated. The stomata is also not differentiated into spongy and palisade mesophyll. There is no differentiation in mesophyll. So, all mesophyll cells are exactly the same. There is no distinction. It is not differentiated into spongy and palisade mesophyll cells. In grasses, okay, everybody has drawn, right? There is nobody remaining. Okay, so now I can write the points. In grasses, certain adaxial epidermis cells, adaxial means upper epidermis, cells along the leaf veins, modify into large empty colorless cells. Large empty colorless cells. These cells are called bulliform cells. This is a very important question. These cells are called bulliform cells. Okay. So, what do they do? Yeah, I hope you have written. When the bulliform cells are turgid, have absorbed water, and are turgid, the leaf surface is exposed. And when they lose water, the leaf surface will curl up. When there is water stress, when there is water stress, that means water is less, the bulliform cells will lose water. The bulliform cells lose water and make the leaves curl. Make the leaves curl. Curl inwards to minimize water loss. Okay, to minimize the water loss. So if you have to draw these cells will be like this. Large empty colorless cells like this. These are the bulliform cells. These are the upper epidermis cells. Upper epidermis. Yeah. So when they lose water, they will suddenly shrink in size and they will form a crease along the leaf surface. They will form a crease along the leaf surface. So if they form a crease along the leaf surface, the leaf will quickly fold up. Okay. All right, so this brings us to the end of the leaf, root, stem cross sections. Now we have to do the secondary growth. Now we have to do the secondary growth. So please put the heading. Secondary growth. Secondary growth. Okay. So secondary growth. Right. So what is secondary growth? Secondary growth refers to the increase in thickness, increase in girth of the plant. So this is called the secondary growth. Primary growth, what is primary growth? Primary growth is growth in length, the growth in the root length, growth in the shoot length. So both of these are vertical growths, but the horizontal growth, that is the horizontal growth, the increase in the thickness, that is the secondary growth. I will explain. First of all, please write the growth of the roots and stems the growth of the roots and stems in length is called primary growth. Primary growth. Besides primary growth, besides primary growth, most dicots exhibit an increase in girth or thickness. Increase in girth or thickness due to secondary growth. Due to secondary growth. Due to secondary growth. All right. So now we will begin the process of secondary growth. Okay. So there are two things here, the formation of cambium ring and the activity of cambium ring. So for that we need to draw some circles, we need to draw some diagrams. So first of all, the first diagram that I am going to draw, not sure I can draw it on this page, so I will use the next page, and this is also, I will have to increase a bit, wait a second. Okay. So we are going to draw some diagrams now, so pay some attention to this. First of all, we need to draw a transverse section of a dicot stem. So everybody can do this. They can draw a section of the dicot stem, right? So when you draw a section of a dicot stem, you will draw many vascular bundles. You will draw many vascular bundles like this. Okay, you will draw many vascular bundles like this. The inner side is the xylem, the outer side is the phloem. So for the xylem, I am going to draw with blue color. Xylem is by blue color. Xylem is blue and phloem is red. Phloem is red. So this is the phloem. So all the vascular bundles, they are arranged in the form of a ring, and in between xylem and phloem is present the cambium cell. Cambium I am drawing with black. Cambium I am drawing with black color. This central part is the pith. Okay, this is the pith. This is the epidermis. So I hope you remember the transverse section of dicot stem, everybody. Everybody remembers how to draw this transverse section of dicot stem. Epidermis, hypodermis. Hypodermis is made up of which type of cells? Hypodermis is made up of which type of cells? Students, please do not fight among yourselves. I can see. Okay, so I understand there must be some reason for your happiness, but yeah, please try to control. All right. So okay. So the thing is, we draw epidermis, then we draw hypodermis. So hypodermis is made up of which type of cells? Hypodermis is made up of which type of cells? It is made up of collenchyma. It is made up of collenchyma. So if you have any problem, students on the, I believe from the right, it is my third row. Please don't fight among yourselves and please change your seats. Please try to be a bit silent. Yeah, please don't fight. Yeah, okay. All right. So epidermis, hypodermis, cortex, endodermis, then comes pericycle in between just behind the phloem, and then there is lies the whole pericycle or sorry, the whole pith. Okay, and between the vascular bundles is a tissue we call the, it's actually parenchyma tissue only, but we call it adjunctive tissue. Okay, so that is also basically parenchyma cells only. So what I am trying to draw your concentration to is this cambium, which I am going to label now. Let me choose a color for this. I will choose green color for this. This cambium, the black colored band that I have drawn, that is the intrafascicular cambium. Intrafascicular cambium. Intrafascicular cambium. Intrafascicular cambium. So this cambium is present in all vascular bundles. Shillong students, I can see you. The students at the back are so silent. At the front, please don't argue so much. I believe your conversation might be disturbing a bit. So if you have to talk, please talk silently. All right. So this is the intrafascicular cambium. This is the intrafascicular cambium. All right. And the thing is, this intrafascicular cambium will help in secondary growth. It can, it has the meristematic cells. It can form new xylem and can form new phloem. But there is a problem. This cambium is incomplete. It is broken. It is patches. Like it's going to be present on the vascular bundles only. So if I am going to draw it like this, it's in the form of patches only, like this, like this. So if this cambium undergoes secondary growth, do not draw this. I am just trying to explain you. If this cambium undergoes secondary growth, we will get the shape of all trees would be like this. If this was the case, but it does not happen. Why? Because now this cambium is going to form a cambium ring. So there is a part here which is played by some parenchyma cells. I have to do some labeling now. So everybody knows xylem, everybody knows phloem, epidermis, cortex, primary phloem, primary xylem. This is the primary phloem, primary xylem, okay, primary phloem, primary xylem, okay, like this. Now the cells between the vascular bundles, they will convert into cambium cells. Now my question to you is, do you remember the process by which these cells will now convert into the cambium cells? You remember the process? Do you remember the process? Yes? Do you remember the process by which parenchyma cells will become cambium cells? It's called de-differentiation. I told you there are three terms: differentiation, de-differentiation and re-differentiation. Do you remember? Yes or no? Nobody remembers. I think I taught you this. Yeah, now everybody is serious, so now it looks better. Okay. Okay, so yeah, you can be happy, but just try to be It's normally only going to be a half an hour from now. That's all the time I have right now. Okay. So there are three processes. I'll remind you, there are three processes. First is differentiation. Differentiation means a meristematic cell, a primary meristematic tissue converting into primary permanent tissue. Do not write this. This is just for your remembering. This is differentiation. Dedifferentiation means a permanent cell converting backwards into a meristematic cell. But meristematic cell which is present from the initial stage is called primary meristem. So we cannot call it primary meristem cell. A permanent tissue cell converting into meristematic tissue. So what do we call it? We call it secondary meristematic tissue. Are you getting me? So this is what is happening here. A parenchyma cell is a permanent cell. It is converting into cambium cell. Okay, a meristematic cell. So this is not present from the start. It is formed later between vascular bundle. Now this will be called interfascicular cambium. Why inter? For example, there are some inter-school quizzes, inter-school competition, inter-school football tournament. Inter-school means between two schools. Intervascular means between two vascular bundles. The place that is present, that is the interfascicular cambium. Intra means within itself. So within vascular bundle is intra, outside is inter. So now in the next diagram that I am going to draw, I am going to label this interfascicular cambium. If there is a problem, please let me know. I have already discussed differentiation, dedifferentiation and redifferentiation in last class. If there is a problem, let me know. So now this diagram is almost going to be the same. I am going to draw a slightly larger circle. A slightly larger circle. Okay, this is a problem. All right. A slightly larger circle than previous. Okay. So there the cambium ring was incomplete. Now my cambium ring is totally complete. Now my cambium ring is totally complete. I have drawn only few vascular bundles here. 1, 2, 3, 4, 5, 6. So actually in your book there are more, and normally it is more only. So that's not much of an issue. Now I am going to draw larger more vascular bundles. Actually in this previous figure I draw only 6, so that you can understand. There will be more than 8 vascular bundles normally in dicot stem. 6 to 8 is very minimum. Normally there are more. So now I have drawn 8. Do not get confused that vascular bundles not increasing in number. If you are getting confused, I will draw 2 more vascular bundles here. Now it is 8 here also. Okay. Do not think that vascular bundles will increase. Vascular bundles will not increase in number. They will remain the same. So now this cambium that is developing in between, it is called interfascicular cambium. Interfascicular cambium. Interfascicular cambium. Okay. So this is the thing here. Epidermis, cortex, primary phloem, vascular cambium, so many things. This is the interfascicular cambium. Now, what is the cambium ring? This part, this was the interfascicular cambium. Okay. Intra plus inter forms the complete cambium ring. intrafascicular cambium plus interfascicular cambium plus interfascicular cambium equals to cambium ring. This is the complete cambium ring. Now I can also call it cambium ring. This is the cambium ring. Now the cambium ring origin is defined as secondary because it is formed from one primary meristem and one secondary meristem. So I will call it secondary meristem. Whole cambium ring is secondary meristem, and everything that is formed from secondary meristem is called secondary growth or secondary permanent tissue. Secondary meristem. Cambium ring is the secondary meristem. Okay. Hope you have finished it. Now try to understand. Before the cambium ring was formed, there was only primary phloem, primary xylem. Now the cambium ring is complete, so uniform layer of secondary xylem and uniform layer of secondary phloem will be formed. Secondary phloem is formed outwards and secondary xylem is formed inwards. Okay. Secondary xylem will be pushed inwards and plant will increase in the overall thickness, overall size, circumference. Circumference will start increasing year by year. More and more xylem and phloem will be formed, and as it is formed, it is going to be pushed outwards. Okay. So let's draw this quickly. Now we are going to draw a larger circle like this. Yeah. Now it is done. Epidermis, cortex, hypodermis, everything will be pushed outwards by the new secondary phloem that is being formed like this. So now what I can do is I can draw a uniform circle now. Primary phloem is being pushed inwards, so I can draw it like this. This is primary phloem, and great amount of secondary tissues have been formed. This is the cambium ring now. It is growing outwards, and as the cambium ring is growing outwards, it is pushing phloem outwards. So phloem is being pushed outwards. This is the phloem, okay, and the xylem is being pushed inwards. This part whole is the secondary xylem, and these are the medullary rays that I am drawing with the blue color. So let's label it. This is the secondary xylem. This is the secondary phloem. Okay. Secondary phloem. This is the cambium ring. This is the cambium ring. And these are the medullary rays. These are the medullary rays. Okay. All right. So let's continue. Now we have to draw even bigger circle. Everybody has drawn it, right? It's not a complicated diagram now. Now what we have to draw is something like this. We have to actually draw a new layer. After much amount of secondary growth, there will be a change in the plant. There will be a new change in the plant. There will be a change in the plant. And I am going to discuss this change in just a minute. Just a minute, like this. Okay, so phloem, phloem. Let me label these things first. This is the cork layer, phloem. I'll explain what it is. This is the cork cambium layer or phloem. Phloem. Cork cambium. You can also call it cork cambium. Phloem is the cork layer. Cork or cork cambium, and then there is a secondary cortex, but labeling it would be a bit tricky. Okay, like this. So this whole part this is the secondary xylem. This is the secondary xylem. This is the secondary xylem. Okay, let's draw it now. Label it. This is the cambium ring. Cambium ring. Outside the cambium ring will be the phloem. The secondary phloem. Secondary phloem. Okay. In the center part, this part, it is the primary xylem. This is the pith. Pith is getting crushed. This is the primary xylem. Now I'll explain this diagram now. So finish drawing it, and I'll begin explaining it. A lot of things to be explained here. Medullary rays I have not labeled. Medullary rays, let's label it. These are medullary rays. Okay. So this will require a bit of an explanation. So I'll do that. please finish it. Look, the thing is, if you compare it to a dicot stem, it will be easier to understand. Okay, I think most of you have finished. Yeah, I can see the pens that you are putting down. All right. Let me continue now. So look what is happening in the plant. In the tree that is undergoing secondary growth, large amount of cambium is cutting cells. Cambium is cutting cells, and cambium is pushing the newly formed xylem cells towards inwards. Okay, you keep on looking at the diagram. Cambium ring is complete now. So cambium will form new xylem. Xylem is formed inwards and phloem will be formed outside. Why? Because cambium continuously cuts cells. It forms two types of cells. Cambium, what does it do? A cambium cell like this, cambium cell, cambium cell. This is a cambium cell. It forms new xylem cells inwards by division. Inwards, or the circular part, inside towards the central part. Towards the outer periphery, it forms phloem cells. This is the outwards or periphery region. Periphery region. So if you focus, you will be able to understand secondary growth, both cork secondary growth and normal secondary growth in just one step. So please give me 5 minutes with full concentration and I'll explain everything. So please focus on the diagram also. Keep on looking at the diagram. Remember the layers: epidermis, epidermis, hypodermis, cortex, endodermis, and vascular bundles and pith. This is the arrangement. Now the vascular bundles have joined with each other. They have formed a large ring, and this is called the cambium ring or the secondary meristematic ring. Cambium will form new phloem, new xylem, because it is a meristem. It will form new cells which will redifferentiate into secondary xylem and secondary phloem. Phloem forming outwards, xylem forming inwards. So xylem is being pushed towards the center, phloem is being pushed towards the outer region. And one more important thing, amount of xylem formed is very much larger compared to phloem cells. So what will happen to the plant? Plant will develop cracks on the outside. Any tree that you have seen, eucalyptus tree, teak tree, shisham tree, neem tree, every tree that you have seen in your life, you must have seen some cracks on the surface, on the bark, on the outer surface. Why do they develop cracks? Because of secondary growth, because xylem is formed in more quantity, phloem is formed in lesser quantities. So that is why their skin, their outer skin will start developing some cracks. Is that clear? Okay. So when you develop cracks on your skin, what do you do? When you develop cracks on your skin, what do you do? In winters are coming, right? So when you develop cracks on your skin, what exactly do you do? Come on. Moisturizer, exactly. So plants have a moisturizing cream as well. They have a creamy substance, and this creamy substance is called cork. They manufacture cork. What happens is, in their outer skin there is cortical layer, cortex. Just below the epidermis is the hypodermis and cortex region. Cortex has parenchyma cells. Parenchyma cells form a new kind of cambium. It is called phallogen. They form phallogen. Phallogen forms phloem. Phloem is called cork. It is going to form new cells which have a fatty substance called cork cells, and these cells will fill the spaces where the cracks are happening. Why do they need to protect it? Because if they do not protect these cracks by sealing it with cork, plant will become infected by pathogens. If the skin of the plant is exposed, fungi, bacteria, viruses, they will immediately attack every plant in which secondary growth is happening. So that is why cork is formed in each and every tree. But there are some trees which manufacture large quantities, very large quantities of cork, and that becomes useful for us. So there is a plant called Quercus suber, and this produces large amount of cork. It is also called cork tree. So this tree, it's a oak tree, and oak tree is called Quercus suber, and that is used to manufacture cork. We scrape the outer skin by a sharp blade or something, and we take cork out, press it, and make so much variety of substances from it. Okay, so that is how cork is made. Also, one more question that I have for you. So while the secondary growth is happening, if xylem is formed more, what will happen to the pith? If xylem is formed in so large quantities, what will happen to the pith of the tree? Will it survive or not? Will the pith survive or not? It has such a huge amount of pressure. So will the pith survive or not? Will the pith survive? Yeah. Actually, the pith gets crushed. Pith is consisting of basically parenchyma cells. So the xylem is going to push it. It is going to crush it. So it will actually get crushed. In an old tree, it will get crushed by the pressure of the xylem. Also, one more thing. Now, before disappearing, there is one more thing that is happening. The cambium does not only make xylem vessel and phloem sieve tube companion cell only. It also makes phloem parenchyma. It also makes xylem parenchyma. Parenchyma, I told you, are living cells. So these are present in between xylem vessels adjoining. So when cambium is making some new cambium is making something like new xylem and new phloem, some parenchyma cells will also be formed. So these parenchyma cells, they help in forming some line of conduction horizontally, not as vertically. They help in lateral conduction of water. So these are called medullary rays. So besides xylem vessels, there will be some cells which will form a line like structure, one first cell, second cell. So each cell is placed one after the other, like students, you stand in a line in the front of the principal every morning. So similarly, cells are lined one after the other. Parenchyma cells are formed continuously. So these are called medullary rays. These are living cells. Now what happens to the pith? The pith gets crushed, and the xylem, primary xylem cells that are present there, they also get filled with some substances called tannins, essential oils, some other substances. Okay, and everybody knows about katha, or everybody knows about sandalwood trees. How many people of you have seen sandalwood trees or used sandalwood tree wood in any form? I use sandalwood tree a lot. Okay, so have you used sandalwood tree wood? Yeah, so it has a very sweet smell coming out of the wood. That is an essential oil, and that oil develops after 20 years of secondary growth. So when a tree is 20 years old, then it is fit for cutting. If you cut it before that, you will not get any sweet smell out of it. So that is because the inner part has been after 20 years, there are some essential oils filled in that primary xylem and pith portion. It will get crushed also, but there will be large amount of substances present in the center. This is called heartwood. This is called the heartwood. Heartwood is darker in color. So now when you go to purchase some sandalwood, what color wood do you, how will you choose which wood is better? Like you cannot exactly tell by smelling, because smell accumulates over a large period of time. It's very difficult to smell. You will get only the smell when you actually try to rub it on a stone mortar or pestle. So how will you understand it? Look, when the tree is undergoing secondary growth after many years, the central portion, it will undergo darkening due to development of tannins. Tannins, essential oil, resin, so many substances will start filling in the center. Because of this change, the central portion of any plant after secondary growth, it becomes darker in color. It becomes darker in color. Suppose this is a tree and I am observing it. So what happens is, after some time, just a second. After some time, the central portion becomes brown in color, dark brown in color. Everybody has seen this, right? Central portion becomes darker in color. This is called heartwood. Please do not write this. I will explain this in the next class, because I have to give you some notes also. Heartwood, and the outer portion is called the sapwood. So this darker color wood, that is filled with tannins, resins, oils, etc. Yeah. So when you are going to buy some sandalwood for yourself, choose the darker colored. Darker colored will give more scent, more essential oils. If you are buying the lighter colored, the smell will be lesser. It will not be that much effective. Okay. That is how you going to. This is how study of biology is going to help you in your daily life also. Okay. So sandalwood, always try a. If you have many choices, always try a wood piece which is darker in color. Darker colored sandalwood piece will give better smell. Okay, it's more scented. That is how you buy chandan. Darker is better, and lighter one, that won't give any sufficient quantity smell. Always a darker one is better. So hardwood, sapwood is seen in most plants. Like if you see an old tree, we'll see this also. One more thing you will see here, there are some rings. I have not drawn here. There are some rings here. There are some rings in the tree. So these are very large rings I have drawn, large spaces in between. But actually these rings are very close to each other. So I'll explain how these rings are formed. These rings are called annual rings. Okay, and annual rings, why do they form? Look, there are two things for a plant. There are two conditions. These two conditions are, in every year there is one favorable season and there is one unfavorable season. During the unfavorable season or unfavorable condition, the xylem vessels that are formed in unfavorable condition, okay, they will be smaller in diameter, because plant is under stress. So they will form very thin walled, thin diameter xylem vessels. And when favorable season will return, it will form larger diameter xylem vessels. So darker, the smaller will appear darker, and larger diameter xylem vessel will appear lighter in color. So dark, light. Every year there will be one dark pattern followed by light pattern, one dark pattern and one light pattern. So when we see this from a distance, what we will see? We will see rings, because cambium ring is a ring. So it will form wood in a ring fashion, and these rings, they will be one dark and one light pattern forms one ring, and one dark and one light ring is called annual ring, because they will be formed in period of single year. Okay, so these are called annual rings. So I believe if I start giving you the description, it will be a bit problematic. I'll continue this in the next class that we have. Because of some issues, my class has been a bit shortened. So from this part I will continue in the next class. Okay. So I have to give you some notes also, but all of that will not be possible in 10 minutes. So I'll take your leave now. Okay, we'll meet in the next class. Okay, so I hope you have drawn it. Okay, so yeah, I'm keeping my word. I told you I'll leave you at 3 o'clock. So after this, we'll meet in the next class. We'll discuss. Okay. All right. So just a bit. So everybody has drawn it. There is no doubt. If you have any doubts, you can ask me on the group. I have to give you some notes also in the next class. Let's see how much we can do. Okay. We will finish this chapter in the next class. Okay. All right. So that's all for today, students. Thank you for your time. Thank you for your patience. We'll meet in the next class. Thank you.